Hello viewers, in this lecture video we have discussed about microbial growth curve. Introduction of microbial growth curve. The increase in cell mass and size that occurs as an organism develops is referred as growth. A bacterial population is number of live cells grows with time, and the growth curve is separated into 4 stages: lag phase, exponential or log phase, stationary phase, and death phase. Growth curves that are often used in biology, ecology, finance, and economics. The first stage is the lag phase, which occurs when the bacteria are not dividing but are metabolically active. The exponential, also referred to as a log phase, is characterized by rapid growth. In the stationary phase, growth comes to a standstill when the number of dying cells equals to the number of dividing cells. During the death phase, the number of living cells decreases exponentially. The bacteria or viable shells should be seeded onto sterilized broth and incubated under an appropriate growth curve in microbiology to examine the bacterial population growth. Bacteria are unicellular organisms with only one cell. The bacteria consumes the part of the medium and expands it in a mass and size. The bacterium is said to be active growing in microbiology. Researchers can study the growth of bacterial dynamics by plotting the log of the cell numbers versus time, or growth versus incubation time. The microbial growth curve are measured with the spectrophotometer. Lag phase. It takes some time for a microorganism to adapt to the new condition when it is first put into a new media. When cellular metabolism speeds up and the cell expands, bacteria are unable to replicate, which results in a decrease in mass of cells. The period of the lag phase is related to the organism's preceding state of growth. It takes the microbes longer to assimilate to its new surrounding when it is introduced into nutritional poor medium. Exponential phase. During this phase, microorganisms multiply and divide rapidly. Their metabolic activity increases and binary fission, DNA replication begins in the earnest. Generation time is the amount of the time it takes the bacteria to double in size over the particular length of the time. The period of time it takes for an organism to reproduce varies. Stationary phase. As the bacterial population develops along the microbial curve, it consumes all of the nutrients in the growth medium. In the medium, toxic metabolites, waste materials, and inhibitory components such as antibiotic accumulates. This alters the circumstances of the medium, such as temperatures and pH, making bacterial growth more difficult. The rate of the reproduction of the bacteria reduces until the number of cells undergoing divisions equals to the number of cells dying. At the point of the time, the bacteria stops dividing completely. The growth rate remains constant because the cell count is not increased. When a stationary cell is shifted to a new medium, it can quickly switch to the exponential phase and restart to metabolic processes. Death phase of microbial growth curve. The bacterium's capacity to move the death phase is aided by the loss of nutrition and consequent accumulation of metabolic waste products and other toxic components in the medium. During this time, the bacterium's capability to reproduce is completely lost. Bacteria difficultly start to die when they are exposed to the conditions that are unfavorable, and the death occurs quickly and uniformly. This is the picture of microbial growth curve. First stage is lag phase, second is exponential growth phase, third is stationary phase, fourth is death phase. Conclusion of microbial growth curve. Bacteria require specific conditions to grow, which are not same for all bacteria. Many factors influence the growth curve in microbiology. Additional factors to take into account include atmospheric pressure, osmotic pressure, and moisture availability. The time it takes for bacterial population to double, also known as generation time. It varies by species to species. I hope now you all understand about the bacterial growth curve. Thank you.